Hi, I'm Ben, and I'm super excited to be able to stand up here and present some of the work that we had funded by the CFRI, particularly in a rough funding environment. So we're going to split the talk. I'll start off, give like a little intro on what we do at the Phage Center, and then I'll pass it off to Katie for the last half. So, because we were debating whether she'd stand up here, but nobody knows what to do with their hands, even me up here. So, anyway, yeah, happy to be here. Hi to everyone online, if anyone's there. And I will try to speak slowly because I have a horrible habit of speaking way too fast all the time. And I get, oh, also thank you to the organizers, since I might not get the chance for the acknowledgements at the end. Especially Siri and Sabina, who definitely kept us organized through all this. So thank you for organizing the conference. There we go. So Katie and I both work at the Yale Center for Phage Biology and Therapy. And it's really like we do basic translational and clinical work all in sort of one institution, which I think is pretty cool. And it falls into three sort of broad categories of research: discovery, development, and deployment of bacteriophages. So like in the introduction, we really do the full like bench to bedside, but also with environment to bench to bedside, where we're out in the field collecting samples, looking for new phages, bringing those samples into the lab, characterizing these phages, and then sometimes using them in the clinic or in, aquaculture, agriculture, a vet, hospital, something. But at the university, it's really cool for me, at least, in that we have the university associated hospitals, so we can actually treat cases and then also like collect samples and just walk them over to the lab and analyze them. And I think that's really cool. So like I said, there's three bits of our research at the university. And, we often talk about new phages at conferences. We have a cool sequence or like, a neat host range, receptor trade-off. And we do, like I said, clinical work where we quantify the clinical impact. But today we're going to talk, or Katie's going to discuss some of the development stuff that we do, which it's really nice to be able to speak at a conference like this and sort of shine a light on that sort of work. So I hope you guys enjoy that. We'll be talking about phages, which I guess I realize I haven't discussed in the intro, which are the viruses of bacteria. And they're like the most numerous organism on the planet. And this is just a quick animation of like a very generalized life cycle of a lytic phage. And you guys are probably all familiar with it, but I want to make sure we're all on the same page because it's pretty critical to the work that we do. So phages as viruses of bacteria out in the environment, they're non-modal, so it's just Brownian motion. They're bouncing all around, encountering bacteria. And the first step of infection is they attach to the host. So they like grab on, and then they inject their nucleic acid or their genome and make copies of it. And then from those copies, they make new particles and then assemble them all together, fill them with their genome. And then the last step is they burst, and then those particles all go on to repeat the cycle. And critical to a lot of our work is the first step, attachment, and also, phage binding, which is surprisingly complicated, I'm learning, in that it requires the phage particle here to recognize a specific receptor or receptors in the environment. And like, it's easier to think of it in the lab, how it's just like, bacteria and phage, but in like the wild, there's these phages, they don't move, so they're on their own. So they're bouncing around in the environment and they're hitting, debris, they're hitting other phage particles, they're hitting many different bacteria species. And so they have to be sure they find the right species to infect because they're not generalists, typically. And so as they're bobbing around, bouncing on the surface of bacteria, they have to recognize specific receptors, which aren't necessarily present on all members of a species or a genus, or we have some that like infect really, a strain or a small subset of a strain within a species, which can make things complicated, I guess, because they're super, super specific, right? And this host specificity for a long time sort of held the development of phage therapy as like a anti-infective back, because you'd have to know a lot about the infection and you'd have to have a huge panel of phages in order to deploy them. But I think now that we're appreciating the microbiome and like the role of a lot of these bacteria in health, I think they're actually now is an opportunity for us to develop them as like a really neat personalized approach to treating infections. So like I said, the attachment part is critical to our work and this like hyper specificity of the phages. And here's why. So if you think of bacteria and phage interaction, it's the life cycle I just showed, happens all the time in the environment. Just as with any sort of anti-infective, whether it's phage or the chemical antibiotic, eventually in this system, bacteria evolve resistance, right? That's why we have the antimicrobial resistance problem as is. And a very easy, I guess, easy for me to say, but easy way for bacteria to evolve resistance to phages is by adjusting the surface receptors. And so we try to exploit this and direct the evolution of bacteria to reverse antibiotic resistance or attenuate virulence. And we see it sort of as like a bit of a spectrum here where, I mean, it's kind of a stupid cartoon, I realize, but like on the left, where we have these bacteria, these really, nasty ones that are virulent and antibiotic resistant, but they're also phage sensitive. And then on the right, they're phage resistant, but they're, they play a little bit nicer. So we try to view it this way and then sort of capitalize on this possibility so that the therapy, if we're fortunate enough to use it, either kills the bacteria outright with the assistance of the immune system and antibiotics, or the bacteria evolve resistance to the phage, and then it opens up the possibility that we can use other antibiotics. And so, so we try in lab to find phages that focus on these virulence factors. In the wild, phages can use really anything that's presented on the surface of a bacteria, some of which is not important for causing infection. And so we really try to narrow our phage selection down to find phages that use, for example, something like this, like biofilm formation or gene exchange or motility. And that's been our sort of core philosophy in how we develop our phage-based stuff, whether it's for treating lung infections or joint infections. We've treated quite a few prosthetic joint infections or implanted devices. And we've also worked in like agriculture and aquaculture. And so we're sort of all over the place, but we still try to focus using the same sort of philosophy. And maybe more relevant to this community is just in a small subset of some of our pulmonary cases. I think we just published this kind of recently. We saw, following this philosophy of treating with targeting virulence factors or stuff involved in antibiotic resistance, a 7.3% average FEV1 percent predicted improvement after, I think, 3 weeks, and a 3-order magnitude drop in bacteria density in the sputum in CF. And more important, I think, for me, was that they were reporting a lot more improvement in quality of life, which is why we're doing all this. And that's sort of what we do at the center. And then I can hand it off to Katie now. Cool, but I guess I'll be here for questions, if there are any.
Well, thank you, Ben, for the intro to Phage and what we do. I'm going to talk mostly about the project that we specifically had funded by CFRI, which is kind of looking at optimizing different types of phage therapy. So typically, and historically, I think people kind of default to cocktail phage therapies where you're mixing multiple different types of phage together and giving them in a treatment all at the same time. And we tend to either use single phage, so like a monophage therapy, or single phage applied sequentially. So maybe 3 different phage applied one at a time over the course of a week. But a lot of this is kind of just historic bias in the literature, and there hasn't been a whole lot of, I think, evidence to back up like which is better, a cocktail or a sequential phage therapy. And that was the main goal of this project. And so to kind of start us off thinking about it and how to design our experiments, we wanted to think about what are the main goals of any antibacterial treatment. And so the obvious primary goal is going to be to eliminate the source of infection or reducing clinical load of bacteria. And a secondary goal that we should always consider is kind of prolonging the time to treatment failure. So slowing the evolution of bacterial resistance to the therapy, whether it be antibiotics or phage. So in the case of an antibiotic, this might mean adding like a secondary molecule like beta-lactamase inhibitor when you're treating with a beta-lactam antibiotic. But for phage, this to us means that you would perhaps want to use 3 phage for treatment, but then be strategic about which order you're going to deploy the phage to make sure that the bacteria maintain phage sensitivity as long as possible. But after training in an evolutionary biology lab for the past 10 years, I have come to appreciate the adaptive potential of bacteria, and they seem to be capable of evolving resistance to pretty much everything. So we typically like to think a few steps ahead if we can when developing therapy, as Ben mentioned, and we think about trade-offs between phage resistance and bacterial virulence, or phage resistance and antibiotic susceptibility. And one of the final goals that we think about is, when possible, to reduce or control the adaptive capability of the infectious agent, so in this case, bacteria. So when bacteria become resistant to the phage, this typically happens because they're evolving and accumulating mutations that confer phage resistance and potentially lead to an increase in genetic variability. And if and when possible, we would like to minimize this. So in thinking about this experiment, design like using a phage cocktail or a sequential phage as a treatment, we chose to start with these 3 phages that we have characterized in lab. The first one is phage 4, which is a myovirus that uses some of the core sugars of lipopolysaccharide, or LPS, as a receptor. The second phage we chose to use is 10A2, which uses the O antigen of LPS as a receptor. And the third phage is 31B2, which is a KMV-like phage and uses the type 4 pilus of Pseudomonas as a receptor. And as a way of kind of starting this experiment, or as a kind of our experimental design, we started with a single ancestral colony, which went through 3 days worth of passaging. One of the treatments was a no phage control. So the bacteria was just passaged for 3 days in a high nutrient medium, LB. And then we had different lines of bacteria that saw the same phage over the course of the 3 days. We had 6 different lines where we gave the phage one at a time in every possible sequence that we could. And then finally, we had a line that saw the 3 phage cocktail. So the phages were mixed equally and were given at the same time at every passage. And so these passages were 24 hours long, and every day the bacteria was diluted 1 to 100 in new media with a constant concentration of phage. And we performed this experiment in biological triplicates. So we started off with 3 different ancestors, and we also did this in 4 different bacterial hosts. So PA01 is the kind of canonical lab strain of Pseudomonas that we like to think about and use in lab. And then we did it also with 3 different clinical isolates that were isolated from pwCF. And for the sake of time today, we're just going to mostly go over the PA1 data, and at the end, we'll kind of compare the results to the results of all the other strains. So one of the first things we measured at the end of each passage was bacterial density by plating out populations with dilutions. And I'm going to show you a lot of these graphs here are the three from the monophage therapy, where on the y-axis we have CFU per mil, which represents bacterial density. And we have on the x-axis time in days. And we can see here that over time, the three different monophage treatment lines didn't have much of a reduction in bacterial density as compared to the control, indicating that likely phage resistance evolved pretty quickly. In the 6 different sequential phage treatments, we do see a 2-log reduction for some of the sequences, but interestingly, not all of them as compared to the controls. And then finally, in the cocktail, we do see a very large reduction on day 1, but the bacteria are able to very quickly recover to near no phage control levels by the 2nd day. And if we look at this kind of more generally, it was this bold change in bacterial density at the end of the experiment compared to the control. We see a significant reduction for 3 of the sequential treatments, as well as the cocktail treatment. And so the next thing we chose to look at was resistance and cross-resistance to other phages that we were planning on using. Cross-resistance happens when a bacteria evolves resistance to a phage that's infecting it. And at the same time, this resistance or this confers resistance to another phage that it hasn't yet encountered. So this would be problematic for phage therapy, as you wouldn't want to use a phage that selects for a high level of cross-resistance to many other phage, as that would limit the choice of phage that you could use in the future. So if we look at the resistant patterns for the monophage therapy, the bacteria we have changing in time on the x-axis, and on the y-axis, we have the phage that the bacterial populations were tested against. And we have this scale here where the darker the shade, the more resistant the bacteria is to that phage. So if we look at just this first sequence here, which was bacteria seeing only monophage therapy from phage 4 over time, we see at the beginning that the ancestor is very sensitive to phage 4. And after its passage in phage 4, it very quickly becomes resistant. And this resistance also confers cross-resistance to phage 10A2, which these bacteria never saw. And we see a very similar pattern for 10A2, which makes sense because both of these phage use different pieces of LPS as a receptor. And then a little surprisingly, I'm sorry, for the monophage therapy for 31B2, we see, again, very quick resistance generated to the phage, but this also then confers a high degree of cross-resistance to the other 2 phage, which these bacteria never saw. For the different sequential phage treatments, the cross-resistant results are kind of dictated by these patterns that we saw in the monophage therapy. So for example, this first one here, which is the bacteria being treated first with phage 4 and then 10A2 to 31B2, we see that we start out very sensitive to phage 4 and quickly generate resistance, which then also generates resistance to 10A2, but then the bacteria remain sensitive to 31B2 until they actually see 31B2 in treatment. And unsurprisingly for the cocktail treatments, they pretty much immediately selected for resistance to all the phage that were in the cocktail. So to summarize these results, we kind of see some cross-resistance evolving in the monophage and cocktail treatments, not as much in the sequential treatments, and all the resistance seems to be dictated by the results that we saw in the monophage treatments. Next, we looked at trade-offs between phage resistance and bacterial virulence, and typically in our lab, we like to use these 2 molecules to assay for virulence of Pseudomonas. So the first is elastase, or production of elastase, which is an enzyme that Pseudomonas produces and degrades elastin in the lung environment. And piocyanin is a molecule produced by Pseudomonas that contributes to a lot of oxidative stress or oxidative tissue damage. And we're able to fairly easily measure these in the lab. So we can extract these from cultures, measure them, and this is what we see. So we see here that elastase activity is very significantly decreased for 3 of the sequential treatments as well as the cocktail treatment, especially compared to the control. And for pwCyanin, we see a significant reduction in expression for the control, but none of the other treatments summarized here. And after looking at the virulence, we looked at a trade-off between phage resistance and antibiotic resistance. We chose 10 different clinically relevant antibiotics and measured MICs to each of these antibiotics for each of the endpoint populations. And they're presented here as a fold change in MIC as compared to the ancestral MIC. And while we don't see any reductions in MIC really for any of these treatments, we do see a few significant increases for some of the sequential treatments. And lastly, we did whole genome sequencing of the endpoint populations for all the different treatments. And through variant calling, we're able to identify polymorphic sites in each of the genomes and we can use that data to then calculate a statistic called Tejman's D for each gene that are under selection. Tejman's D is a statistic commonly used in population genetics that quantifies the observed nucleotide diversity in a population under a few different assumptions. And a positive value here is indicative of balancing selection, which is characterized by a decrease in population size, both with low and high levels of polymorphic sites, while a negative value is indicative of positive selection, which is characterized by population growth and a large number of rare variants or low frequency polymorphic sites. So basically anything that you see that is below 0 in this kind of green, blue, purple color is indicative of a larger number of genetic variants being present in the population. So here we've mapped the polymorphic sites in this heat map. And on the top, we've quantified the total number of genes that are under selection for each of these populations. And we see that many of these populations have levels similar to the control here at the end in black. One of the sequential treatments had a very, very low number of genes under selection. And the cocktail treatment had a higher number of genes under selection. So we see, or this indicates that populations, especially in the cocktail treatments, have an increase in genetic diversity as compared to the other treatments, which is perhaps not what you want if you're going to be treating someone with phage. These results are kind of recapitulated here in these 2 graphs, looking both again at the same statistic, Tejman's D, and then nucleotide diversity to look at different genes that are undergoing the most selection. And these are kind of a preliminary analysis that we'd like to look at more closely to investigate specifically which genes are kind of contributing to this likely phage resistance. But in the end, really what we wanted to know is which treatment is better. And maybe not unfortunately, but not surprisingly, it kind of depends on what your goal of treatment is. So we kind of outlined at the beginning, reducing bacterial load, minimizing cross resistance, reducing bacterial virulence, reducing MICs, or minimizing genetic variation. So it really depends on kind of what you want as a goal of the treatment. So if you want something that only has better outcomes as compared to the no phage control, maybe you want to go with the 3rd sequence, which reduces bacterial load and minimizes cross resistance and reduces bacterial virulence, but we didn't see any change in MIC, and we didn't really see a big drop in genetic variability. If you really want to limit genetic variability, then maybe you want to go with this sequence 5, where we saw limited genetic variation, a large reduction in bacterial load, reduced production of elastase, but a slight increase in MIC to piperacillin tazobactam. Or maybe you really want to reduce bacterial virulence. So you choose to use the phage cocktail, which reduced bacterial virulence and reduced the bacterial load, but we didn't see, we actually saw an increase in genetic variability. So it's very important to consider kind of the end goal of treatment when choosing which therapy to use. And unsurprisingly, it also really depends on your bacterial strain that you're trying to treat. So these are the same results that I just showed you for PaO1. And these are kind of the results summarized briefly for the other 3 strains that we used in this experiment. And when we repeated it, we saw that the strain CF19 had results that were fairly similar to PaO1, but the strains CF21 and CF31 were pretty drastically different from PaO1. So while this may seem kind of like bad news, that there aren't kind of a single set of rules to dictate how we should be treating people with phage, and this historically has been kind of viewed as one of the major drawbacks to phage therapy, is that you need to know a lot about what bacteria you're going to be treating and what phage you're going to be treating with. We see this as kind of a really great opportunity to focus on tailoring phage treatment to specific strains of bacteria to kind of personalize this medicine for each person that we're considering treating. So in summary, the choice of monophage, sequential phage, or cocktail phage treatment really depends on the goals of treatment, the targeted bacterial strains, and though we didn't specifically test that here, likely the phage that you're planning on using for treatment. And in the future, we're really hoping to incorporate this type of, well, this experiment similar to this type of experiment when considering treating someone. So we're working to develop kind of procedures to perform these experiments that are in higher throughput with a larger collection of phage from our phage library. So that in the future, we can do experiments like this for each person that comes to us seeking phage treatment. And with that, I would like to thank the CF community and the CFRI for funding this project, and thank everyone at the Yale Center for Phage Biology, especially Paul Turner and John Koff, who along with Ben are the directors of the center, and Emma Kane and Manu, who kind of piloted this experiment.